how this book is. Jump all over the place. Okay. 533, landmark discovery. Read first.
Me.
Okay, Light Day, go ahead.
533.
533, landmark discovery. Extraordinary new technique changes biology forever. 533.
San Francisco, California, 1973. Both the study and practice of biology were radically and irreversibly changed with an announcement by Stanley Cohen of Stanford University and Herbert Boyer of the University of California, San Francisco. They reported the first successful and a recombinant DNA molecule. What makes this accomplishment so remarkable is that the recombinant DNA molecule made by together or recombining pieces of DNA from two completely different sources never existed before. Today it exists, but it exists in many thousands of copies, and researchers can make thousands more at will.
Okay, so recombinant DNA, recombinant is we're going to take two different things and, DNA from two different organisms and put it together. Recombine it. Okay? This is sort of like the basis for some of the gene therapy. Okay, next paragraph. Richard.
One congratulate on their accomplishment. Darwin and Boyer quickly acknowledged that their depth to many institutes hard work and important made creating and cloning a recombinant molecule possible. One key advance in just a few years was the 1970 discovery of the restriction enzyme by Warner Arbor of the University in Basel and Daniel Nathans and Hamilton Smith of Johns University. Restriction enzymes are proteins that recognize and cut perfectly a specific sequence of nucleotides in DNA.
The restriction enzyme is what's cutting the DNA that's allowing them to recombine DNA from different places, okay, from different sources. And they have to do this. This is sort of like the basis of the gene therapy, okay? And the enzyme, remember, is made from the protein. How's the protein made? What decides how the protein is made?
The genes.
The genes or?
DNA.
DNA, right? The DNA is the directions for the protein. Okay, next paragraph. Dominique.
Another key advance took place in several research projects. That discovery involves I study another important protein, the enzyme DNA ligase. DNA ligase functions during replication and repair DNA molecules. It repairs links that occur in one DNA strand. Isolating DNA ligands allows scientists to use it to fasten back together DNA fragments that restriction enzymes have cut. In addition, the DNA that scientists stitch together can come from different organisms. For instance, scientists can combine bacterial DNA with DNA to create recombinant molecules.
Why would they want to put bacterial DNA together with yeast DNA?
To make a new organism.
Make what? Make a new organism of like half bacteria, half yeast?
A new type of DNA, maybe?
A new type of DNA? Make what bigger?
The DNA.
Make the DNA bigger?
Don't yeast rise.
Well, yeast, the yeast doesn't rise. The yeast is an organism that when it eats sugar, it gives off carbon dioxide, and then that's what makes the bread rise. So you were thinking it would make the bread rise even more. Okay. Okay. Richard.
Because it was saying earlier in the paragraph that it repairs nicks.
Okay, you're thinking maybe the bacteria can repair nicks in some DNA. Okay. Okay. We'll see. Next paragraph. Any volunteers? Keela, go ahead.
That didn't have time, but the technique was straightforward.
They cut DNA from two different
We just read that part.
recombinant
Recombinant.
DNA techniques or revolutionizing scientific research and technology. Scientists around the world are isolating and inserting a wide range of human genes into bacteria. By doing this, they are able to both study gene expression and harvest the products that the genes code for. Such techniques may lead to an unlimited supply of human insulin in treating diabetes or restricted quantities of human growth hormone to use in treating dwarfism. Imagine inserting these for a wide range of desirable characteristics into crop plants, bigger cucumbers, frost-resistant strawberries, sweeter corn, who knows? One day we may even be able to remove particular genes from people's bodies and replace them with other genes.
Okay, what do they mean by harvesting products from the genes? What do you think that means?
Maybe taking certain traits from the gene.
Traits? Okay, like what traits?
Like, okay, I guess, like, alright, like, I have blue eyes, but maybe I want brown eyes. They could take a certain gene out that would take the blue eyes away.
Why would that be useful? You're going to harvest. What do you harvest now? What are some things you
Or
harvest
they
now?
fruit.
Fruit? Vegetables. Okay, so it should be something that's benefits somebody, right? So what would they want to harvest? Sarah?
Maybe there's like a cure for some illness to some DNA, and if they harvest that special trait, they can put it in another person for the.
Or I guess they could harvest the I guess a part of it that has to do with a disease.
Okay, if we're talking about humans maybe in the
Yeah.
future, but
Well,
yeah, you're right. They might take the part of the DNA that makes that particular organism better and combine it with a different, maybe makes it bigger, and combine it with another DNA that might make it sweeter. Okay, something else they can do, we're talking about insulin. They harvest those genes to make more of the insulin from animals like pigs. Okay, so then they can copy that DNA and they can reproduce it and make lots of insulin for humans. Okay, so that's how they would use it for animals. Okay, and that's what they mean by harvest. They take out what they want to use. They get it to make what they want. Go ahead.
Of course, new technologies come with potential problems too. Scientists are already reporting out the need to regulate technology to allow only beneficial and safe transfer. The deliberate combining of genes from different species, however, involves controlling genes and evolution in a different way than than has ever been done before. We may not be able to predict exactly the impact that those changes have. Our world is a different place today from what it was yesterday. It is important that all of us, scientists and non-scientists alike, be knowledgeable and take part in deciding how we will use our new genetic abilities.
Okay, so we have to think about these things, okay? If we change something to try to make it better, how is that going to affect our society? Okay, how is it going to affect the rest that we didn't change?
We'll make it safe.
You'll make it fake.
Like generic, because we already, it's kind of something, you want to make it how you want to make it, it's going to make it like not all right. It's going to be like all those steroids and stuff like that. Is it like how they do it food, how they put all them steroids to make it bigger, how they want it, so it's going to make it fake.
Okay, so one concern might be what they're putting into it that might not be so good for us.
Would they be creating something like a Jurassic Park in some way?
That's a good question. Do you think that could happen?
Yeah. If you put your mind to it, I think they could. 'Cause they were already trying on the video we watched. They were trying to make a dinosaur with that mosquito they found in the blood they found from millions of years ago from the dinosaur.
Just
Okay.
wondered.
Okay, okay. Do you think they're going to recreate a dinosaur someday? What do you think? They tried with the chicken, right? Richard?
I think so, yes, because, how it's going now, I think that if that if they had the chance to have a live species rather than just the samples of the blood and things, I think they would definitely create it so that they could get more of an aspect of how it lived much better.
Okay, so you're looking at it through the in terms of you're gaining more knowledge about how the dinosaur lived, and we can be more specific. We've not.
I would also have to say yes, because you gotta think, if they're doing this little DNA extraction thing where they can isolate it and make it bigger, then I guess they could do that to maybe the mosquito, make the DNA, like, copy it, manipulate it and things, and send bigger surges of the DNA into, like, the chicken or whatever animal they're trying to create into a dinosaur. So I guess it could work.
You should. Okay, Sarah.
I would like to comment on what Akila said. The reason that it didn't turn out so great was because they said there was so much DNA within the mosquito from it taking all sorts of things that it got mixed in with the dinosaur DNA, so it didn't. Okay.
So the DNA from the humans that were trying to do this experiment got mixed in with the DNA that they were trying to insert for the dinosaur, and what could happen then? Yeah, we don't know what might happen, right? We have
And
no
then we probably
clue.
trying to make a mate for that one.
Okay, okay.
It's like Mary Shelley's friends.
Okay, we have one more essay we need to get through very quickly. Manipulating genetic material.
What page?
And this is page 534. Okay, so that's what I gave you an overall view. Okay, this essay they're gonna talk about some specific things they do to manipulate the genes. Okay, and then your groups are going to be talking about what this means to us and what this means to society. Sheila. Oh, go ahead. First paragraph.
In the last 40 years, science has greatly increased our understanding of the structure and function of genetic material, as well as our ability to work with it in the laboratory. In fact, our increasing ability to study and manipulate molecular processes in a variety of species has led to a revolution in the ways that scientists conduct research. That, in turn, has changed how
Pharmaceutical. You know what that is? Pharmaceutical. That's companies that make drugs, that make medicines and things like that for you, called pharmaceuticals.
That, in turn, has changed how pharmaceutical firms make medicines and how many industries conduct their day-to-day business.
Okay, Sharina.
All of the technologies are new are fundamental. Interest in studying and manipulating genetic information is not new. For centuries, humans have selectively bred plants and animals to produce organisms with desirable combinations of characteristics. Traditional methods of agricultural breeding were the earliest form of genetic engineering. Genetic engineering is a process designed to artificially control the genetic makeup of an organism.
Okay, so just stop there for 1 second, because that's the topic that we're looking at today, genetic engineering. Okay, so that's the idea that you can selectively choose how you're going to engineer an organism or change an organism or breed an organism, that humans choose what they're going to do. Okay, that's going to be our topic today. Go ahead.
In selective breeding, humans cross plants or animals that have desirable traits to produce new generations. For example, a golden retriever is the product of selectively breeding various Labradors and
Spaniels.
As a result, future generations show more of the desired traits, and less desirable traits sometimes can be eliminated.
Okay, hold up right there. Has anybody ever seen a golden retriever? Anybody have a golden retriever? Okay, really nice, gentle dogs. They're really nice, calm. They make nice pets. Okay, and so that's a pretty good dog, right? So that would have been something good. Go ahead.
For instance, a greater tendency for developing hip
Dysplasia,
dysplasia, a common cause of
osteoarthritis,
or
degenerative joint disease.
develop in many selective dog breeding programs.
Okay, so there's a downside to it. Those dogs that they breed have problems, okay? Hip dysplasia, that's problems in their back hip, and it makes it really hard for them to walk, and they suffer a lot of pain, and sometimes they get broken hips, broken bones because of it, okay? So they make really nice dogs, but on the other hand, maybe you're breeding in some of these problems, okay?
That's what happened to my aunt's dog. I don't know what kind of dog it was. It was like a golden retriever, but black.
Okay, the
So-
lab, probably a black lab.
Yeah. So that's what happened to her.
Okay, yeah, and it's pretty painful for the dog when that happens. Okay, Jesse.
In recent years, scientists have developed more powerful techniques for examining genetic material at the molecular level. These techniques make it possible to selectively change the genotypes of organisms. Many other new techniques allow scientists to alter the genetic information of a species in much more direct and extensive ways than the older methods of selective breeding. It is now possible to introduce genes into an organism that neither parent possesses. It is even possible for scientists to remove genes from one organism and introduce them into an unrelated organism, one that does not normally possess those genes. The new DNA formed through that process is called recombinant DNA.
Okay, so they can introduce these genes, like all kinds of different genes that didn't even come from the parent, okay? They can put genes in there from all kinds of things, and it recombines, and then hopefully, after it recombines, what might happen? Why would they do it? Why would they put all these different genes in there? What do they hope is going to happen after they put it together and combine it? Richard? Okay, that's why they do it, to breed in these desirable traits. But how's the trait going to become expressed? They put the DNA in there, and then what do they hope happens?
It,
changes something that was wrong with it before.
Well, maybe it wasn't even wrong. It just changes it.
Better.
Maybe want to make it better. Okay? But then how does it end up being like something that the next cucumber has, is bigger? How does that happen?
Well, because it'll help it'll help the cucumber, and so the next cucumber will get that, and it'll just become evolution, and it'll just change and
Okay.
it'll pass on.
Okay. That's exactly what I was getting at, what Daryl was talking about. Did anybody else get that?
So pretty much what you're saying is that the gene will change something in the animal, and then it'll pass that genetic mutation down to the other.
Exactly. And then you end up having a whole population of that, organism or that cucumber that you were trying to breed. Sarah? Sarah, you have a question?
So basically, scientists are messing with evolution.
That's a very good point. You hold that for your discussion, your group discussion. Okay? Very good point. Okay? Possibly. What, I don't know. You're going to let me know. Okay. Who wants to read? Daryl, go ahead.
One example of how scientists are using recombinant DNA technologies to address a specific practical problem involved the cotton plant. Cotton plants often are attacked by a pest called a bullworm, which damages cotton crops and costs millions of dollars each year to control. Researchers, however, have known for a long time that bullworm will die if it eats the common bacteria called
BT. You can call it BT.
Okay, BT. For short. Okay.
Okay, so this cotton plants grow. Anybody ever seen a cotton plant?
Yeah.
Okay, it's like little balls of cotton. They grow on the plant, and down south they have these bullworms. Okay, they're about this big and they're pretty fat, and they get in there and they eat it, and they're really a pest. Okay, if you grow cotton, these are really a pest. Okay, so they found out that if this bullworm eats this bacteria, it'll die. Okay, and that's where we are.
I picked cotton before when I was riding back from the beach, and it was just this field. It was just a plantation basically, so no houses or shacks or nothing, and it was like real like the cotton's like this big and it's kind of rough or whatever. It's not as soft. But then you see some and it's just some are not there, got holes and stuff.
That's from the bullworms.
Yeah.
Yeah, eat it. Oh, wait. Got it. Just wait for it. Okay.
The protein that bacteria produces is partially digested in the worm's gut and poisons the worm. Because that protein is so effective, for years farmers have sprayed PT bacteria on their cotton crops just to discourage the bullworm from eating them. The protective measure is that it draws back through sunlight. Sunlight breaks down the BT, and rainfall easily washes it off the plants.
Okay, so if the rainfall is washing it off the plants, where's it going?
And the soil.
Okay, anything wrong with that?
Yeah.
Plants will eat it for energy. It'll get the moisture out of the soil. And it's like some BT. It will splash off the rain. You put it in the ground with a whole bunch of water. Fill some of the water up to its roots and eventually die.
Okay, that's one, could be one drawback, right?
If it gets in the cotton plant, wouldn't it be enough load?
There was another idea, okay? Hold that thought, okay? Keep those thoughts, yes.
Like if it gets when it gets in the soil, the plant's sucking up. The plant is being poisoned as well as the cotton. And that's not
Okay.
bad because we use cotton for all types of stuff. Like most of our clothes is cotton.
Okay,
And
so you
stuff
think
like
that might transfer to you?
somehow it could break out your skin or
Okay,
do something.
okay, all right, hold that thought. Okay, we don't have much more to go, guys. How about line A, how about you read? Through genetic engineering, 535.
Through genetic engineering, however, researchers have overcome those drawbacks. Figure E12.6 how scientists isolated the gene that codes for the poisonous bacterial protein and transformed the gene into the cotton plant. The new cotton plants thus contain recombinant DNA, and they are able to produce a bacterial protein, BT toxin, in their leaves. Those plants are engineered to protect themselves from damage because when a ball worm begins nibbling on the leaves, it eats the BT protein and dies. Food crop plants also can be engineered to produce the BT protein.
Okay. Keep going.
Because it is not toxic to humans. A benefit of such technology is that farmers can use less insecticide, decreasing the amount of toxic chemicals that enter the waste supply of food web. Technology, however, has not been completely effective. Farmers using the engineered cotton in 1996 reported that many bollworms survived in the new crop.
Okay, hold on 1 second. How were they able to survive the new crop?
They got used to it.
What happened? Yeah.
Like something's been happening for a while, like the same thing when we get vaccinations. Like they put a little bit into us, and then our immune system gets used to it and everything.
Well, I don't think cotton has an immune system.
Well,
What
no,
do you think
the
happened?
bullworms.
The bullworms? I don't know that the bullworms have an immune system. What might have happened to the new generation of bullworms? Sarah? Well, not immune, that's not the right word. What happened? Some sort of mutation must have occurred that made them resistant to this toxin, okay? Just like in those scenarios, remember we did the scenarios with the viruses? Could have been similar to that, okay? Then what might we end up with? If we start getting bullworms that are resistant to toxins,
Very good, and it's going to have the same problem all over again because they're going to be the plants is resistant to this toxic thing now, and they mutated into just dealing with it. But now the bullworms, they're I'm not saying they got a brain or nothing, but they're going to start getting mutating. They're going to start growing the
bullworms
custom
are getting better, right? Stronger, better, maybe bigger, I don't know. Okay. Go ahead. I'm thinking like monster bullworms, right?
Additional pesticides needed to be applied. Though the total amount was reduced, surviving bollworms raised concerns, however, about that prolonged exposure to BT.
Other organisms altered by genetic engineering techniques can be just as important, however, even if they are not transgenic. Consider the genetically engineered tomato, which recently was approved for sale in the United States. Tomatoes normally ripen and soften at the same time. That situation encourages farmers to pick the tomatoes while they are still green and ship them to stores before they soften. The less desirable alternative is to ship ripe tomatoes that are easily damaged in transit. Unfortunately, green tomatoes do not ripen. Well, after they are picked and often have little flavor, even after they have turned green.
Have you ever eaten a genetically engineered tomato? How do you know?
Well, because my grandma grows her own tomatoes.
In the winter?
So, well, actually, yeah, she has little tomato plants for the Enzyme.
I don't eat tomatoes.
I do.
Nobody eats tomatoes.
I
I eat lots of tomatoes.
I don't know, like, I eat ketchup, but I don't, like, I'll eat them sometimes in salads, but I'll tell my mom not to.
And I have no idea whether or not the tomatoes I'm eating are genetically engineered.
You can probably tell because of the size sometimes. Because I know like organic stuff is smaller than the food we usually eat now. Like
Yeah, does it have to be labeled genetically engineered?
Yeah.
Genetically
No.
engineered is different
it's
than
just how it
organic.
is now.
Okay, so you really don't know if you're eating it or not, do you?
I don't eat tomatoes, but
I eat tomatoes like I eat apples, like that.
Yeah, I like it too. I eat a lot of tomatoes.
that's
So it
really
makes me
good.
think. It makes me think. Okay, Amy, next paragraph.
But wouldn't it taste different?
Amy, we're at the bottom of 537.
Wouldn't it taste different?
I don't know, because I'm not sure if I've ever eaten one. What do you think?
I think we probably tell the way it tastes.
Okay, Amy, bottom of 537, first column. When scientists.
When scientists discovered that the ripening and softening processes were not the same, a new alternative for farmers emerged because different genes and thus different biochemical pathways regulate the two processes.
You can keep going.
I lost my look at F. Scientists were able to target the softening process and modify it. The result was a genetically engineered separation of the ripening and softening pathways. This allows tomatoes to be ripened on the vine, preserving their flavor, yet transported to the market without damage and rotting. Farmers and consumers alike benefit from that new technology. Some questions remain, however, about the potential effects if a genetically engineered crop is interbreeds with a wild type of crop. Those questions cause scientists, politicians, and citizens to weigh the benefits and concerns for technologies carefully.
Okay, so they were pretty good. They could sort of pick out, a certain kind of gene that made the tomatoes firmer. Okay, didn't soften so quickly and ripen not as quickly. And they put them into the other tomatoes that they were going to be delivering. Okay, and that made the tomatoes better. But what if some of that stuff they put on there accidentally mixed with the crops that were just growing on their own? Okay, like they didn't put it in where they wanted it. Okay, because they put it in a certain place. They put it into the DNA in a certain place. What happens if they get mixed in? Okay, we don't know.
Or it can make them worse.
Right, we don't know. That's part of what you're going to actually be talking about today. Okay. Next paragraph. Akila.
Genetic engineering also has a great effect on human health. Insulin, the peptide hormone that diabetics require, at one time could only be obtained from the pancreas of cattle and hogs. Insulin produced from these animals were available in limited supplies and it was expensive. Furthermore, insulin from other animals was not very effective for some individuals because it was not similar enough to human insulin. By recombining the human insulin genes with bacterial genes, however, researchers have been able to produce human insulin in large bacterial fermentation systems.
Mm-hmm.
This process is an economical method for producing authentic human insulin.
Okay, so they sort of like, cloned it and kept copying it. So they get lot of people believe that. That's why there's all this controversy over the stem cells having to use the one line that George Bush said, you know, we can only use this one line that was already there. Because after you copy it so many times, some of the researchers believe that there could be defects in it. Yeah. Amy.
What is the, I don't know how to ask this question, but what is the purpose of why they want to clone things in people so bad? Like, do they really have a reason, or is it just something they want to do?
Well, for instance, what you just read about the insulin, okay, what we just read about the insulin, they can only extract that or harvest it from live animals, okay, like hogs or pigs, and that's insulin that people who, like, are diabetics, they need that every day. And so if they can sort of clone the part that they need, they can make it artificially. Okay, so that's one thing. That's one use. But cloning, you're talking about cloning as far as like Dolly the sheep. Okay, and that was just probably, you know, for research purposes to see if they could do it. Okay, and Dolly survived a little while. She had a lot of problems, and she died young. So, but they do have success, like when they're working with DNA to map the whole human genome. They had to do what's called PCR, or use PCR, which is sort of like cloning, to get a bunch of pieces of it to study, to be able to know what was on each of those genes. So that's a good use. If there's a crime scene and there's only a little drop of blood, okay, they need to copy that. Okay, and that's sort of a form of cloning or copying. Not exactly cloning, but use PCR for that. They need to make a whole bunch of it so everybody can look at it and study it, because you don't want to damage the original one, right? So they make a whole bunch of copies to look at it. Okay.
I forgot.
No, Zach's going to read the last paragraph. Did you have a question? Not right this second. How about read the last paragraph for us? Researchers.
Researchers currently are working on ways to treat certain human genetic disorders. They use recombinant DNA that has been constructed from harmless forms of viruses and functional copies of human genes. Introducing genetically engineered human cells into the human body for the purpose of curing a genetic defect is called gene therapy. The first successful attempt to accomplish gene therapy began on September 14th, 1990, when genetically altered white blood cells were introduced into a circulatory system of a 4-year-old girl, Ashanti De Silva. Ashanti was born with
Adenosine deaminase.
In this condition, an abnormal allele of ADA gene fails to the scientists hope
No,
that
you skipped the whole thing.
a key protein is required for the correct functioning of the T lymphocytes, cells that are critical to individuals' immune response. The absence of this protein meant that Ashanti, like other children who would inherit ADA deficiency, would easily die of infections that would scarcely trouble other children.
Okay, so they're doing human trials now. So the first one, this is Ashanti De Silva, it worked on. Okay, it worked really well for her. So good outcome. They were able to use it for some other people. Okay, then they tried, and how they do this? Okay, they take bacteria, okay, different kinds of bacteria or viruses, okay, mostly viruses, and they put some kind of DNA into the virus. How often do viruses reproduce in bacteria?
And then it was every minute or every second.
Like 20 minutes or so, right? Okay, so they put these into the human, and because bacteria and viruses, the DNA is just sort of floating in there, it's not encased in anything, they can get at it real easy. So they put some DNA in there that they want to introduce into the person, and then they inject them with this virus. Okay, so the virus goes to your cells, right? It attacks your cells, and then it gets into the cell, embeds itself. Then as the cells reproduce, that virus with the changed DNA is copied and sent on. So that's how that works. Okay, they've done other studies this virus to another person, and he had a bad reaction to it, and he died. Okay, so some were good, some were bad. Then they had to stop the trials, okay, because he died from that. And that's what you're going to read on that last page there. If you're doing humans, you're going to want to go back and read over some of that. Okay, and they call that germline therapy. We talked about another time. What if they, I mean, it's one thing to introduce these viruses into a gene that's going to reproduce your hair or your skin or something like that. What if they introduce it into an egg or a sperm cell that's going to actually change a new person? Okay?
But I remember what I was going to say. If they, about the insulin, if they would have never started, like, creating it to how they want it, would it have help diabetics better? Like, isn't insulin supposed to be, is it like a cure or something for diabetes?
It's not a cure. There's no cure for diabetes, but it's a treatment.
A treatment that makes you,
They
that
don't
makes
produce
it.
the insulin they need, so they have to take injections of it.
So do you think that would have helped them better? Like, maybe if it was just pure and not genetic?
Well, it is pure because they're reproducing it the same as what it was. It's being copied exactly like it was. Okay, so the rest of this, if you are the group who's talking about humans, you'll want to look over these other trials that are on this last page. Okay, go back to page 472. Okay, now let's look at number 3 on page 472. We're all talking about genetic technology, okay, or genetic engineering. There's, and you know, whenever they want to do any sort of genetic, or working with genes, or working with something, they have to look at what's called, they have to decide what's ethical. What is ethics? What do I mean by ethics? Richard.
What's human treatment? Like, appropriate.
What's appropriate, okay? What are some things you need to consider when you're making an ethical decision?
How it's going to affect you in the long run.
How it's going to affect you in the long run, right? What else?
Probably wrong. I was going to say how it affects others, but
How it might affect other people, right? Okay, how it's going to affect you, other people, Richard.
How it might affect your environment.
What it might do to the environment. Okay, all really good questions. Okay, so if you look at number 3, A, B, C, D, E. Lide, read A. 3A. Four, 7, 2. How about you can read B? Sharina, read A.
Okay. What do you use information?
3A.
Okay. Use the information from your science essays and any other
No,
reliable.
3A, huh?
Okay. Identify and summarize at least 2 major ethical issues related to the technology associated with the topic.
Okay, so when you get in your groups, in the 3 groups we're going to have, one of you is going to talk about genetic engineering in plants, one's going to discuss genetic engineering in animals, and one's going to discuss genetic engineering in humans. Okay? So you're going to talk about, identify and summarize 2 major ethical issues related to the technology associated with genetically engineering those. Okay? B.
Explain at least one benefit or one concern related to each of the major ideas that you summarize in step 3A. Be sure to include discussion
Dominique.
Describe how the issue that you displayed have already affected or might affect you and others in the future.
Okay, D. Here.
Explain your opinion with regard to the balance between benefits and concerns that you identify and expect to be.
And E. Daryl.
Recommend what you think should be done about the issues that you have summarized.
Okay, so there's the 5 questions that you're going to consider when you get in your group. Okay, so again, we're going to end up with a roundtable discussion. Okay, so first you're going to start discussing this in your group. What are some things you need to know for your group?
One, you need to know your topic. Yeah.
Okay, so what is your focus question?
Which is pretty much 3A and 3B, right?
Okay. Number 3, A to E. What page is that?
That's 472.
472. Okay. What else should you remember when you're in your small groups?
Your topic.
That's what that
Well,
just
let's
said.
see. The topic is genetic and
Oh well.
To
You have
take
a focus
notes.
on it, don't you?
That's what this focus is.
What about inclusion?
I'm talking about when you're discussing this. What are some things you got to remember, Sarah?
To make educated ideas and guesses instead of something that's really ridiculous.
Educated ideas and guesses? That sounds like the scientific method. You're discussing this. What are some guidelines you need to have?
State your opinion and if you have a different opinion, then discuss it out.
Be respectful.
Okay, respectful. Respect others' opinions. Okay? What else do we need to remember? And this is actually for our small group and our large group.
Don't interrupt when others are talking.
How else could we put that? Be polite, be respectful, polite. How else are we going to put that? If you're not interrupting when somebody else is talking, what are we doing?
Listening.
Okay, we need to listen. Who's talking? How many people? Okay.
Don't put me out.
I need one person talk at a time. And what are the other people doing?
Listen.
Taking
Listen.
notes.
Taking notes. Try to reflect on someone's statement.
Right. Good. Listen. Take notes.
Thank you.
Reflect. That's a good one too. Okay. We need anything else on our list? Can we do it from here? Okay. What else? What's our big thing that we're trying to get skilled at here? We keep
accountable.
accountable. Very good. Okay. Accountable talk. Right?
Amen.
That's what our goal is. Trying to get skilled at that. You guys are getting pretty good at that. Give me an example of accountable talk. Just a couple.
An example of accountable talk would be using, I know, but like I can't.
If I said something that you really don't agree with.
Accountable talk, like if you said something I didn't agree with, accountable talk would be like, I disagree with you because such.
Okay, that's a good example of it. Okay, what if I really disagree with you?
I highly disagree with you because.
What if I agree with you?
I agree.
Sarah?
Agree or disagree?
Okay,
Agree.
and you're just going to say I agree with you?
No, you have to state the reason why you agree with them.
Yes, I want to hear reasons, okay? Give good reasons. That's why we write these essays, okay? So you have something to back up your statement. You want to be able to back it up, right? With evidence and reasons. You want to be able to persuade somebody to come to your point of view, right? Okay? So this time we're going to do something a little bit different when we're done with our large group discussion or our roundtable discussion. We're going to do what's called a 1-minute paper. Okay, and if you look over here, I have written 3 things, and you're going to have 1 minute to respond. I'll tell you when. What was the most important or useful thing you learned today? What 2 important questions do you still have? What remains unclear? What would you like to know more about? Okay? You got to do those 3 things in 1 minute. And you don't have to write the question, just the answer. Okay. All right, so let's see. How many people do we have?
20.
20. Okay, we're going to have 3 groups, so 2 groups is 7 and 1 is 6. Okay, who would like to do plants?
Can you tell us all the topics first?
Plants, animals, and humans.
You know everybody going to do
Well, we can. So, okay, Daryl wants animal. Well, plants can be, remember the bullworms and the tomatoes. There's lots of stuff in there about plants. Sarah, Richard, Sheila,
I'll do plants for today.
Sharina, Monty, and well, wait, Devon? Okay. Okay, animals. Sky, is that Sky?
No, that wasn't. I was just.
Like everybody in the whole school. Because that's my whole life.
Like people like.
3A through E. It's the 8th.
Those are the questions. Okay, John 472.
We have plants.
These are
Guys.
Oh, you guys don't even open focus point.
You told me to stand.
Oh, right. Monty staying on question. Sarah, you are too. Sure, Richard, turn back to the question. We'll make people on after. No, he's on the question. No, he doesn't have to. Trina is right on that page. It's 532.
You with the story?
Anyways, guys, for the topic,
So you keep your.
I think we should write genetic engineering and then put like a little hyphen or something and put plants so they know that we are plants, not animals or humans.
Well, you're doing genetic engineering of humans. Okay, so it's in both of those 2 essays that we just read, which are these 2 right here. And these are your focus questions right there. So the first thing you need to do? Identify and summarize 2 major issues related to technology associated with your topic. So 2 major ethical issues related to genetic engineering of humans.
1, 2, 3, 4, 5, 6. I'll be the group leader. Please. I think I'm good leader if I try. Okay. Anyways. So, alright, our questions? No.
You were talking about genetic engineering of humans. Okay? That would be the last page. Okay. 534 past 530. Yeah.
Alright, so there's 5 questions. Who wants to double up on them? Like that we can have like 2 people double up on a question because there's 6 of us and we all have to answer.
Okay, one person reads the question and the rest of us.
If you were going to do genetic engineering for some sort of animal, okay, what are some things that you'd have to consider? Pros and cons. Think of it like pros and cons.
5 people read a question, and 1 person just doesn't read a question.
Okay.
All comment.
Oh, okay. Alright, so Richard,
You can think of it as like pros and cons. Okay? What's the good thing? No, genetic engineering of humans.
Richard,
Okay?
Monty, C, Sam.
Well, it means like manipulating the genes for a human. They're, taking out a disease, like gene therapy kind of thing, yeah.
If you remember,
Yeah.
what letter your question is.
So what's just a Gattaca?
It could be something like, removing a disease gene, it could be,
Well, we're all changing
just change eye color or change height or whatever that is.
Instead of plotting, can we actually
About human genetic engineering. Okay, what are some pros and cons? What are, yeah,
So what's good about it? What's bad about it?
exactly. What are some things that could go wrong? Yes.
A lot of things because you're altering your body.
There you go. That's what they want you to discuss. Use those
hold on,
questions as your focus.
because D
So just go through each question and sort of everybody put out your opinion.
D sort of should just go to B. Technically, there should only be 4 times we read a question.
Issues related to our
Explain at least one benefit. Explain your opinion.
So what would be one benefit of genetically engineering a plant? Any kind of plant, pick a plant.
Well, if you grow larger, you can cost man world hunger.
There you go, there's a benefit. What would be not so good?
It could have bad reactions to the system.
Get poisoned.
There you go. Okay, so there's something, there's 2 things there. Okay, so that's what they want you to do, and then just kind of discuss like what could go wrong. Okay, what might go wrong. Okay, so go through all the questions and do that.
Can you help us find something?
Well, they want to know about, okay, the first, okay, so, okay, where are they getting these animals? Okay.
And they're going to be killed anyway.
Okay, so is that good or bad? Okay. Is it, so
Well,
they're,
what if you like mutate it so that it's like bigger? Then you have more birds.
Yeah, you have more.
I think that's a good thing.
so you're saying you can genetically engineer a bigger cow and you'd get more meat from it?
Alright, more cheese.
Okay. Do you want to eat that genetically engineered cow?
Well, our plants are really because they put
Well, you're looking at the focus questions. These are your focus questions, okay? So, you're not on the right page. You're not on the right No wonder you don't know what you're supposed to do.
It's the same question.
Identify your
Anyone need the answer?
So you associated with your topic. So to the technology of genetically engineering animals. Okay, so 2 ethical issues associated with genetically engineering animals. Okay, I heard somebody say something about, you know, what are you going to do with the animals after you test on them? Okay, animal cruelty.
Well, what if they're tested badly and you can't eat them? That's mean.
Okay, so stay with your questions, you know, answer the questions, and then we, you know, and if you're respecting everybody's opinion, remember.
You can't say, this is wrong. No.
Follow the guidelines,
Okay.
Discussion of the issue. Okay, everyone write just one benefit, so right below right below B.
Something good that comes out of it, right?
Basically, yeah, good or bad.
Okay.
So,
It's just the color. If you change the color, like from an endless or I want to know what the
Doors, the
prescription. I mean, it's like regular. Get that little camera.
you can't do that as a group.
You just want to
You're
get
going to go through each question, and everybody say what they think.
You just want them to have a side effect.
Mine
wanted to be able to